What are the latest and greatest in molecular virology? On this episode, we'll be talking about the Molecular Virology Workshop that just happened in West Palm Beach. This is a terrific workshop that is organized by the Pan American Society for Clinical Virology, or PASCV. The workshop precedes the Clinical Virology Symposium organized by ASM, and most people like to go to both. Today we'll be talking about some of the high points from the Molecular Virology Workshop with two of the members of the organizing committee from PASCV. Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. I'm your co-host, JCM Editor-in-Chief, Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. Please subscribe to this podcast and rate or review it. Before I introduce our guests, I have a special announcement. We will be recording 2 episodes of this podcast live at the upcoming microbe meeting in Washington, D.C. On Friday, June 10th at 2:30, Dr. Cesar Arias will be recording an episode on Stenotrophomonas multifilia. And on Saturday, June 11th at noon, Ellie, the co-host, will be talking to Dr. Bobby Pritt. And if Bobby is the guest, you know that parasites are the order of the day. I will be there with Ellie as well. I'll be acting as a warm-up comic. I'll be playing walk-on and walk-off music on a banjo. These live recordings will be in the ASM studio, located in the ASM booth in the exhibit hall. So please join us on Friday, June 10th at 2:30 in the afternoon, and again on Saturday, June 11th at noon. Don't worry if you can't make it, because both of the episodes will be posted in the podcast feed as usual. So I am joined by my co-host and a fellow editor of JCM, Dr. Ellie Thiel from Mayo Clinic. Ellie, how are you?
I'm pretty good. Though honestly, if you want to know the real truth, I feel like I'm still on a post-first in-person after the pandemic meeting high. It was so good to see everyone and really commiserate about the last 2 years. So I'm doing good.
I agree. It was really nice to be back in person. Now, how are you feeling about recording a live episode at Microbe?
So I'm excited. Dare I say that I'm not excited? If you put away the stress of potentially making a fool of yourself on live camera in front of all of your colleagues, it's really exciting. Can't wait. But I'm also not sure if I'm more excited to talk to Bobby about parasites or to listen to your comedic one-liners, So lots of excitement happening.
All right. Well, good luck. I'll try to come up with some zingers between now and then. All right, let's turn to our subject. As I said, we're talking about the Molecular Virology Workshop, and we have 2 guests. First, Dr. Erin Graf. Dr. Graf is the Director of Clinical Microbiology at the Mayo Clinic in Phoenix, Arizona. Erin, thanks for joining us.
Thanks for having me.
And we are also joined by Dr. Stephanie Mitchell, who is the Director of Medical Affairs at Cepheid. Stephanie, we're glad you're here.
Excited to be here, Alex. Thanks for inviting us.
Yeah. So we're going to talk about 3 different presentations from the meeting. And the first of these is one that was titled, Enough is Enough. How much revalidation do I need to do? And this was presented by Dr. Rachel Leisman from the University of Kansas Health System. And Erin, I think you picked this one out for discussion. Can you give us an overview of what Dr. Leisman discussed?
Yeah, thanks so much. I did indeed pick this topic. And for those that are new to MVW or have not been to the workshop before, it truly is a workshop in that it is intensive discussion, very informal setting, centered around practical case-based examples that all of us can relate to in the lab. And so in the mornings, we usually like to have the bread and butter type of sessions. And so this was a great example of one of those. It's something that really impacts all of us, no matter whether you're in a clinical public health lab or an industry partner. So Dr. Leisman gave a fantastic session. She used very specific personal examples from her directorship about the types of validation or verification that she did after things like a move of a piece of equipment, after different types of maintenance on that equipment. And, we kind of unpacked in that session opinions really on what is enough? Because I think the beauty of a lot of these practical bread and butter sessions is it's not an exact science. It's not black or white. There isn't necessarily a right or wrong answer. And so when is enough enough? And, unpacking laboratory developed procedures versus an FDA cleared test, for example. So I personally was excited for that session because my entire lab is moving in about 6 months. And we're moving down a floor through an elevator and through a very large hallway. So, what is enough, I think, is up for debate.
My lab just moved. Anyone who is watching this and has watched in the past can see that I'm in a new office that's still pretty plain because I haven't decorated yet. So I have just been through this personally. But Ellie, if it's all right, we'll start with you. What stood out for you in this session?
So I have to give kudos to Rachel first because she took a potentially very dry topic and made it really relatable with the example she gave. I think one of the questions that was asked was, when do you revalidate when you move an instrument? What do you do if you move it across the hall, or you move it 5 feet away from where it was versus moving it from another floor or another building? And I think through the discussion and in her examples, there was a really nice, basically, she really nicely outlined that, there's a difference about how much validation or revalidation you do based on the extent of your move. And I thought that was really nicely outlined, where you do maybe accuracy and precision if it's a small, short move, versus if you're moving floors or buildings when you're going to want to do kind of a complete revalidation. So kind of talking about mini validation versus full validation, and what is a mini validation. So that was one of the take-homes that I thought she really nicely outlined.
Well, no surprise, I have strong opinions on this, but I'll go last. How about you, Stephanie? What did you take away from this one?
Yeah, I mean, similar things I thought. And, what's great about MVW is that even after kind of the official talk was over, a group of people got together to continue the discussion based off some of the questions and comments that were made by the audience. And, there was, again, the age-old discussion of, well, is it a verification or is it a validation? And what do those terms mean? And like Ellie said, the mini verification or mini validation, people, that's very subjective, and people have very different of opinion on what that actually represents. And so it was really nice to kind of hear from colleagues and others, doing the same thing, and kind of either validating what you're doing is right or getting new ideas of how to approach something. I think one of the other things that struck out to me was, when you have not so much moving of an instrument, but when you have update in software of the device, or a change, like a maintenance change of a piece of equipment within a device, how much re-verification would you need to do in that case? And I think there's still a lot of kind of uncertainty and unknowns around that, because the manufacturer may not provide you as much information that's needed in order to make that judgment call. Sometimes it's just a system function check that's needed that the manufacturer does, and then provides you documentation that's sufficient. And maybe you only run QC. But then depending on what the change is, you might want to do more. And so I started to wonder, as a manufacturer, what additional information can we provide laboratories to help them understand what the changes are, so maybe they don't overdo, too much, when really, maybe just QC was sufficient. So I think opening that question and having dialogue around that was extremely valuable.
That's terrific. And Erin, anything to add? Anything else that stood out for you?
Yeah, I'll piggyback a little bit off of Stephanie's comments. When Dr. Leesman started putting this talk together, she was very vocal about how little information is out there. So there, as I said, it's not an exact science, and there's a good amount on when you bring in a new assay, how to validate or verify. But after moves and maintenance, there is really just scarce literature or guidelines out there. So I think that's room for collaboration, perhaps with our industry partners, to put together some sort of guidelines that can help folks have something to point to say, okay, this is the scenario happening right now. Therefore, I need to do X, Y, and Z to feel like I'm compliant.
Thank you. And we were talking about moves with Erin, your lab moving in about 6 months. And as I said, my lab having just moved this week. In fact, we are still moving stuff right now. So this is a very fresh issue for me. And my take on it is that you validate a test, or the manufacturer validates the test at the initial implementation of that test, or in the lead up to FDA approval for that test. And most of the other things that we talk about when we talk about moving platforms, or when we talk about small changes to assays or upgrades to instruments, I think those are verifications. And you don't need to do a full validation of the assay from soup to nuts. You just need to make sure that it works. So with an instrument that's being moved, if the manufacturer moves the instrument and switches it on and it powers up and everything looks the way it should, you're halfway done. Not all the way done, halfway done. I think that the requirements for running samples on this are pretty minimal. And if anyone from my lab is listening, they'll call me a liar, because I made my people do a lot more than I think is actually really necessary. But anyway, that's my 2 cents. I think really you're just making sure that the instrument is still working. You don't need to do the full validation as you would do with initial use of a test. So. And we can talk about the distinction between validation and verification in another episode when we have a couple of hours to indulge ourselves. The second session we're going to talk about is a debate. And there were a couple of these debates. They were a lot of fun. Had they done debates in the past, or was this something that was new?
So this is the brainchild of Matt Binnicker. We piloted it last year on our virtual workshop, and it just got tremendously positive feedback. It was so much fun. So we had to bring it back again this year.
Yeah, I thought these were a great hit. But Stephanie, you had to moderate one of these, and I assume you organized it and everything. Can you start us off just by talking about how these debates were structured? Because they were highly structured events.
Yeah. So I think, some people might call something a debate in other conferences, and really it's more of a panel discussion. And again, this is all credit to Matt Binnicker, but he got this idea from NPR's Intelligence Squared, which is a podcast that NPR puts out that really is a more true formal debate style, where you have two teams. You have a team for the debate statement, and then a team against the debate statement. And typically it's a very clear-cut statement, which once you get into debate, obviously it's not black and white. We always kind of end up in the gray. But, the whole point is, to really center around what that statement is in its actuality, even if it is black and white, and then have those two teams kind of give their arguments for why that statement is valid and correct or not correct. And so typically we start out with polling the audience first. This is a very interactive session to see kind of, are they, how the audience is feeling. Are they already kind of in the camp of, yes, I agree with the statement, or am I not in agreement with the statement, or am I completely undecided? And so we poll the audience first, and then we actually get into the debate. So we have, bona fide official opening statements from both teams as to their initial arguments, for their stance. And then we basically open it up to questions from the audience, either virtually and in person, and also from the moderator, and really gets the debate going and gets discussion. And there's a lot of back and forth, between the two teams, and also the audience as well chimes in with additional comments or additional kind of follow-up questions. And that takes up the majority of the session. And then at the end, we allow the teams to do a kind of last effort, kind of, again, just, reiterating what their key points are for why the audience should either stay for the motion or against the motion. And then we poll the audience again to see how many people have changed their minds on the topic. And the team who sways the most votes is the ultimate winner of the debate. So it's a lot of fun. And as Erin kind of mentioned earlier in the theme, it's very topics that labs are currently dealing with, that there really is no clear-cut answer, but it drives a lot of discussion because there's a lot of different things to consider. So it makes for really good debate statements. Yeah.
Thank you. And I have to say, and I was talking about this with some other people at the meeting, the debate statements that you chose were absolutely terrific because they really were things where people could get engaged and people would have differences of opinion. And we saw that during the debate. So for the one that you moderated, what was the topic statement and who were the participants?
Yeah, so the one that I had the pleasure of moderating, the debate statement was only certified clinical laboratory scientists should be allowed to perform clinical molecular virology testing. So you can obviously see that's a very clear-cut statement that there is no answer that is clear-cut. And the team that was for the motion was April Abbott at Deaconess Health System in Indiana University, and then Donna Wolk from Geisinger. And then the team against the motion was Susan Butler-Wu from USC, and then Rachel Leisman from Kansas. And they did an amazing job debating back and forth.
They kind of came out swinging. What were the basic arguments that were presented?
Yeah, so they did come out swinging. And I have to say, like, when we think about people to put on these debate statements, we try to find people who not just are passionate, but are willing to kind of throw some punches and kind of get the banter going and take some shots, take and receive shots, right? The whole point of this is to debate your side. And the people that we put on a team may not personally actually agree with the statement. So it's actually even a challenge for them if they're not in agreement to sit there and argue, for a side that they don't particularly agree with. But I think they both made really excellent points that, again, I can see both sides. I think for the debate statement, so Donna and April, I think they made a really good point right off the bat regarding, the value in our profession. We've always been struggling to get the lab, seen, the lab heard, and, really showing that we add value to the healthcare and the care of our patients. And so their argument was, if you start to say that anyone, with any credentials can come in and run laboratory testing, that we're devaluing our profession. And so I think that was a very, very strong statement. And I think it does have some validity. She made some comparisons to the nursing field, where, they are also seeing staff shortages, but they're not really welcoming anyone without a nursing degree to come do their nursing roles. And they're really kind of promoting, the value of that profession. So I thought that was a very good argument from the pro team.
Yeah, they also threw in Theranos and Elizabeth Holmes, and that was kind of a mini mic drop moment. So yes, they had good arguments.
Yeah, they did not hold back. Yeah, no, they did not hold back.
The 6 feet of physical distancing was
necessary
in this situation.
Yeah. I don't know. And then for Rachel and Susan, who were against, the statement, their argument was also very compelling. I mean, they gave a lot of, I would say, they had some data from recent surveys, either by AMP or ASCP, both pre-pandemic and during the pandemic, that showed that, we have been struggling with workforce shortages, and it even got worse, and so we're going in the wrong direction. And, they did say, while there's these lofty goals of, promoting the profession and getting better pay, those things take time. But what happens right here and now is what they're concerned about. And they did have a lot of emotional kind of experiences and examples that they gave that, really makes you go, yeah, I mean, they're having to send out testing because they don't have the staff, or they're having to cut down on their test menu. And how does that impact patient care? So, patient care is on both sides of the argument. But, I think, they were really focused on, well, I have to do something now. what can I do? And so I think one key takeaway that Susan made was that she was really focusing on what steps in the molecular testing process that a non-CLS person could do with appropriate oversight. So really kind of plugging these people in places where their skill set is appropriate and not asking them to do something that, we feel uncomfortable or they're not comfortable doing. And so I think kind of the nuances of where in the testing workflow would they plug in was, I think, a very interesting concept from Susan.
So it was sort of aspirational / idealistic versus practical. Right?
Yes.
That was my take. Erin, what did you make of this one? What did you think?
Yeah, I came in feeling one way and then almost immediately changed my opinion. So I was one of those who was swayed by actually the four. I think that point about devaluing our profession is so profound. And I was really glad I was able to take that away from the meeting because it's perhaps something I had taken for granted, the significance of that certification and us as a profession. We all know that our people are underpaid, they're overworked, and that is a key to getting them the recognition they deserve is making them known and valued as a profession. But on the flip side, I thought it was quite funny. The example that I really enjoyed, I think it was Dr. Abbott during her earlier session had referenced this example of a temporary employee or perhaps it was a research employee that came into the lab to help with COVID testing. And they said, "Can I use the hood in the back? The hoods up here are full." And they said, "Yeah, sure, go ahead." So she took all her primary COVID specimens to the back and pipetted them in what she thought was a hood, and it turned out to be a dead air box. And so, then everyone was terrified that this poor person had been exposed to all these COVID positive samples. And I think this was pre-vaccine availability. So it's just those moments that we all experience where it's kind of that aha moment of, oh, wow, I took for granted that you didn't have this knowledge because you weren't trained in this kind of setting. And we threw you into this without really taking the time to explain the safety risks and all the things that you learn about when you're in a proper CLS type program. So again, I can see both sides of the argument, just knowing the national staffing shortages and the crises that we all faced and what we had to do to get through COVID. So many of us, probably all of us MacGyvering, just patching things together with duct tape and, just saying prayers that it would work. So I definitely see both sides of the argument, but I was very much swayed on the pro side.
How about you, Ellie? Did you change your vote?
I did not change my vote. But I fully agreed and can relate to both sides. Like Erin said, really good arguments made. So for reference, my lab, about 60% of the technologists are CLS. The rest have a 4-year science degree. And, I have to say, I don't see a difference, right, in the quality that they provide. So I think, as was laid out by Susan and Rachel, with appropriate training, people are trainable. With appropriate training, I think we can get everyone to a place that they need to be. On the flip side, obviously, we don't want to be devaluing the profession by any means, but we need a short-term solution, right? We can't just be sending out testing to reference laboratories because, frankly, we too are seeing workforce shortages, and we too are, discontinuing tests because of that. So I think we need a short-term solution with the need to promote the profession and to get a work or a pipeline for additional programs, training programs.
Stephanie, you were moderating, so you were working during this whole session and may not have been able to sort of engage with the content quite as much as the rest of us. But what was your take on this?
Yeah, I mean, I'm with Ellie. I see both sides. Like, I think we have to tag team, we need a short term and, providing maybe laboratories with some fundamental kind of training curriculum that allows them to make sure that they're covering things like biosafety, and, molecular technique and all of that stuff so that they can fill those gaps they have now, but still pushing with all of our amazing professional societies that have been working on this for many years, or have recently made it a super high priority. I know ASM has made this a very big priority of theirs. And so, doing both. I think we can't do one or the other. We really have to go at it from multiple angles to keep the labs up and running, to keep the testing up and running, but, still providing us a future more long-term. Yeah.
Great. And before we go on to the third session, I just want to mention all of these sessions were recorded, and I reviewed them, getting ready for this, and I'm sure you all did too. They're really high quality recordings. So we will put the link in the show notes, and people should check these out, and particularly the debates, which I think were a lot of fun. I'm going to go on to the third session that we're going to talk about, which was the other debate. And this one was moderated by Matt Benniger, friend of the show, has been a guest, and again from Mayo Clinic. And the debate statement here was low or uncommon results should be confirmed by additional testing. And on the pro side, saying that they should be confirmed, were Safani Jean from Nationwide Children's Hospital, Dr. Safani Jean, and Dr. Neil Anderson from Washington University. And on the opposing side were Kathleen Starecht from Albany Medical Center and me. So I will describe the participants' point of view here as well, but I'll just sort of summarize the arguments that we gave in our opening statements. Neil went first, and in support of the statement, he said that repeating low or uncommon positives should be done when it's justified by the clinical impact and in light of what you know about the performance of that specific test. And he made another point that I thought was very good, which was if you're going to do this, you need to do it in a consistent manner following a protocol. So this should not be done on an ad hoc basis, Neil argued, by looking at curves and making ad hoc decisions, because then your lab will be behaving in an inconsistent fashion depending on who happens to be on service. So he said, do this by protocol, do it consistently. Kathy went next, and she argued that low positive results, low viral loads, or low loads of pathogens are part of the natural history of disease. You should expect to see low loads of virus early and late in disease, and that repeat testing on those low loads will not get you any closer to the truth. The repeat that you do has the same limitations as the initial test if you use the same assay, or if you use an alternative assay, you open yourself up to new limitations. And she pointed out that if you try repeating using a less sensitive test, you are sure to get the wrong answer. I'm getting a little passionate because I was on this side of the argument. So then Safani went, and she added to Neil's arguments, and she gave a very strong example, I thought. She talked about a case in which a CMV PCR had a very high CT value, indicating a low load of virus, but the slope was a little bit unusual. And so they dug into it. They repeated using a different assay. And when they did that, it was strongly positive. And it turned out that there was a mutation in the target for one of the primers that had been used in that initial assay. And that's why the load looked to be so low when in fact it was not. Then it was my turn. And I will tell you, I did this debate, and then I also did a talk later in the meeting. I was much more nervous for this debate. I was pretty nervous. I think all of us were kind of spun up about this. But I went for the heartstrings, and I did this because of a debate that I watched or listened to from the NPR blog that Stephanie mentioned, or the NPR podcast, rather, that Stephanie mentioned. So the first argument I made was pretty dry, and that was the consequences of confirmatory testing are twofold. The first is that if you use an FDA-approved assay and you do repeat testing of the same sample and change your report based on that repeat testing, you are out of compliance. You are no longer following the package insert, and so you are doing a laboratory-developed test, and you have to do a full-on validation of that test. And I made the argument that that means that only the biggest labs are able to do this. Only the academic centers, only the large reference labs will be able to do this, and we are essentially taking these tests out of the hands of mid-sized and smaller labs. And then the other argument, and this is where I went for the heartstrings, was the clinical harm that would result from delaying results, issuing erroneous results, and all of these things leading to a lack of treatment for patients' infections. And I had a picture. I fooled around with the pictures a lot for this because we were putting up slides during this. I put up a lot of pictures of I initially had a lot of pictures of cute kids and other people being tested. And I ended up with just one picture that I thought was very moving of an elderly patient being embraced by a doctor in full PPE. So I don't know if that was fighting dirty or not, but I would be interested in what you all thought from the other side of the podium. Why don't we start with Stephanie?
Yeah, Alex. I mean, I thought you were on point. Like, I did not change my vote. I'm very much against the repeating for all the reasons that you guys mentioned. Especially when you're near the LOD, you're gonna get flip flop. I mean, that's just the nature of the analytical limitations around that. So, what is truth, and how do you get to it is challenging. So. But I thought you were not playing dirty at all. I thought you played it well. And, we ran into this a lot, and it always kind of comes down to, why did you send the test in the first place? Like, I know in my past experience, we would often get these, weekly positive HSV, samples on neonates. And, then the lab, they're calling the lab saying, is this a false positive? This doesn't seem right to me. And I'm kind of like, well, then why did you send it in the first place? And, and they're like, oh, well, they weren't high risk, but they were classified as low risk for HSV infection. So we sent it. And it started to make me wonder, well, if you have these two buckets of classification, if you are in the low risk group, how does that actually change how you manage or work up the patient? Because if you're in the low risk group and you still send the test, where you're more likely to question a positive result, then now you've gotten yourself into a tizzy, because it'd be really hard to say, I'm not going to treat an HSV positive in CSF on a neonate, right? I mean, that's really hard to ignore or to justify or explain away. So typically they always ended up treating the patient, but, just kind of, it brings to, more color to this debate statement of, should you really be doing it? Why you send the test? Because as Kathy pointed out, if you're in the beginning or the end, that's where you're going to get these weekly positive samples. And so we have to be cognizant of that, whenever the patient is being worked up.
Yep. And that's great. That's one of the things that came up during the open discussion as well. So Erin, what did you take away from this one?
I came in undecided. I think there are some scenarios in which repeats make sense, and I think I was really framing that around a particular GI panel that is fraught with issues. Not necessarily the fault of the panel itself, but other things related. So I was kind of wearing that lens of, when do I consider repeating? But then as soon as you and Kathy made your arguments, my vote changed. I was really moved by this now becomes off-label use, and that was actually validated by one of the industry colleagues who got up and said, yeah, that they agreed that would be considered off-label use and would require a full validation. I really honestly hadn't thought of that before, which is what is so fun about these workshops is it's these simple little things that you come away with that are just mind-blowing. So yeah, for me, it really was everything that everyone already said. What do you do with those repeat results? Is it an orthogonal method? If you don't have access to that, you're just doing best 2 out of 3. I mean, nobody likes that. That's terrible. I think that was mentioned as well. So yeah, I was swayed.
How about you, Ellie? I bet we didn't sway you.
Yeah, no. So serology, I feel like, is a little different. We talk about why is this positive and this is a low positive, and is it really positive? We should repeat it. I mean, it's like a common everyday question. So it's difficult. I think one of the things that came out from this debate was the importance of communication and communicating with your clinical team, with the lab, and in some cases taking it on a case-by-case basis. So I think that was really important and something that both sides really stressed. As for repeating, though, low positives, we had a situation come up a couple of weeks ago where it was an FDA cleared test, and we started noticing that we were seeing a lot more positives that were in that low positive range. We did send out for testing by a different method, not retesting them on the same method, and they kept coming back negative. So, you get into the question, is this an LOD issue with the assay? Well, we ended up reporting it to the company, who then came back 2 weeks later and issued a total recall of the assay. So I think it is important to investigate low positives. One other point that was made is, yes, they're FDA cleared, but that does not make them infallible. So I do think you need to probe at certain times. So, Alex, you were very convincing. But
we don't have to talk about my part in this.
Well, your team was very convincing, but I am probably still going to be analyzing and potentially repeating low positives.
It raises a good point, and it goes back to something that you mentioned earlier, Stephanie, which was we were given an assignment, right? And I think all four of us in the debate that I was in, and I would wager all four in the other debate as well, probably had some much more nuanced point of view about this. Sometimes yes, sometimes no, here's why, that kind of thing. But with the debate structure, you're sort of pushed to make your points very clearly. And I think that is, I think it's great. It's much more useful for the audience, I think.
Yeah.
Yeah. I think it helps frame the arguments. And, one thing that came up after, which I wanted to bring up with this group, is like a whole nother debate maybe, which was, Neil was saying it should be protocolized, right? Don't do it by case basis, because then you're off label and you're doing all this stuff that could be bad. But one situation that came up is, again, like the example that Sophie gave, which was the curve looked weird, which is different from the curve looks normal. The assay appears to be working as expected, but it's just a weak positive. So clinically, I have some suspicion, or maybe there's some contamination in the lab. But how would you handle something where you see something in the results that makes you go, something malfunctioned and potentially is giving me a false positive or a false negative because the assay is no longer working as expected? In that situation, would it be more appropriate to repeat that because a wonky curve, or, not just is it weak, but it looks weird? I feel like that's a different debate. That's a different discussion. What are you guys' thoughts on that?
Erin, what do you think?
Yeah, I agree. I think that really does highlight the value of having directors like us and then seasoned technologists in the lab who can recognize those things. When you think about labs that are just running FDA cleared assays that are all locked down, that don't even give you the ability to review the amplification curves, I know that's something we all often advocate for. Then you lose that ability to do that analysis that you're describing. And I think the example that Dr. Jean used was their lab developed CMV assay. I know from my time at Children's in Philly, we had similar findings, not on CMV, but other viral assays. And we had experts, seasoned techs that knew that, they knew to look for those flags and bring them to us. And then we could do that really interesting troubleshooting and sequencing and all of that wonderful stuff. So I feel like we lose a lot of that with these very locked down types of assays. So I don't know where the right balance in that is, especially with what we're just discussing about the workforce shortages. And, we're losing that rich depth of expertise that we have had historically.
Yeah. Ellie, anything to add?
Not really. I agree with, what both Erin and Stephanie said entirely.
Yeah, I agree as well with Erin. When you see those funny curves, you have to dig into it one way or another. If it suggests that the whole batch is bad, well, that's just a bad batch, right? And that requires looking into also. But when it's an individual sample for an individual patient, that's when it requires more digging, not only into the amplification and the chemistry itself, but into the patient's chart. And talking with the physicians who are caring for that patient, I think, becomes important as well. Stephanie, to back up, I should have asked you, who won the debate that you moderated?
It was Susan and Rachel. So the team against the statement. Yeah.
Yeah. For the debate I was in, Kathy and I won, but it was a Pyrrhic victory. The other side started out with 60% on their side. We had 20% on our side, and there was about 20% undecided. We pulled a little more of the undecided than they did, but they still had a clear majority. So while we were the victors, we were not the majority vote.
Can I just add,
Yeah.
Alex, that you had one of my favorite quotes of the entire workshop? Something along the lines of "nothing good happens after 40 cycles." I think that's...
I'm thinking of having T-shirts made.
Yep.
Yes.
Yeah.
So before...
I'm going to file that away for the next workshop, Stephanie. Keep that in our back
good,
pocket.
Yeah.
Nothing good happens after 40 cycles.
But then Neil came back and said that he'd argue that nothing good happens after 35 cycles. So I think you can have a front and back
that can be a debate
situation.
Yeah.
It becomes a matter of degree then. But I think basically Neil and I were on the same page. I want to give credit to Rick Rossi, who is a lab scientist here at Children's Hospital. And when I used that line, "nothing good happens after 40 cycles," I told the story that Rick and I were talking about a case just like the ones that we were talking about in the debate. And as we were walking down the hallway, he said, "Nothing good happens after 40 cycles." And I said, "I have to go put that on Twitter right now." Anyway, we should get shirts made. I think
Fantastic.
It's a good one.
Yeah.
I mean, just all sorts of swag, Alex, like shirts, keychains, mugs, just everything. Yeah.
Lanyards.
Lanyards.
Well, thank you guys. This has been an absolutely terrific discussion, and I hope it has given people an appetite for the workshop. I would encourage everyone who didn't get to go this year to give it a try, and you can check it out by looking at the videos, which again, we will put a link to in the show notes. So thank you, Erin. Thank you, Stephanie, for joining us.
Thank you for having us. And I just want to give a quick shout out to the other organizers, Morgan Pence, and then the fabulous Jess Warner from PASCV, who you complimented the quality of the recordings. That is all her. She is fabulous.
Terrific. Thanks for pointing that out, Erin. Ellie, great to see you. I will see you again in person soon.
Looking forward to it.
At our live recording.
Live recording.
All right. Thanks, everyone. Bye now.
Thank you.
Bye.